Hello everyone, and welcome back to Learning Center with Hanif. In today's lesson, we will discuss translation, the second major step of protein synthesis. By the end of this video, you will understand where translation occurs, the molecules involved, the 3 stages of translation, and how proteins are synthesized from genetic information. Let's begin. What is translation? Translation is the biological process by which the genetic information encoded in messenger RNA, or mRNA, is converted into a polypeptide chain, which later folds into a functional protein. Think of translation as reading a recipe. The mRNA acts as the recipe, the ribosome acts as the chef, and transfer RNA, or tRNA, delivers the correct ingredients, the amino acids. By following the instructions in the mRNA, the ribosome builds a protein 1 amino acid at a time. Translation occurs on ribosomes located in the cytoplasm. In eukaryotic cells, translation also takes place on ribosomes attached to the rough endoplasmic reticulum. Components required for translation. Before translation can begin, several important molecules are needed. First is mRNA, which carries the genetic code copied from DNA during transcription. Second is the ribosome, the molecular machine responsible for protein synthesis. It has 2 subunits, a small subunit and a large subunit, and contains 3 important binding sites, the A site, the P site, and the E site. Third is transfer RNA, or tRNA. Each tRNA carries a specific amino acid and has an anticodon that pairs with a complementary codon on the mRNA. We also need amino acids, which are the building blocks of proteins. Finally, several protein factors and energy molecules, including ATP and GTP, help drive the process efficiently. Stage 1. Initiation. Translation begins when the small ribosomal subunit binds to the mRNA. The ribosome searches for the start codon, AUG. Remember this carefully. AUG is the universal start codon and codes for methionine. An initiator tRNA carrying methionine recognizes the AUG codon by complementary base pairing between its anticodon and the codon. Next, the large ribosomal subunit joins the complex, forming a complete ribosome. At this point, the initiator tRNA is positioned in the P site. The A site is empty and ready for the next amino-acyl tRNA. Translation is now ready to proceed. Stage 2. Elongation. Elongation is the stage where the protein grows. A second tRNA carrying the correct amino acid enters the A site. The ribosome carefully checks that the anticodon matches the mRNA codon. If the pairing is correct, the ribosome forms a peptide bond between the amino acid in the P site and the amino acid in the A site. This reaction is catalyzed by the peptidyl transferase activity of ribosomal RNA. After the peptide bond forms, the ribosome moves 1 codon forward along the mRNA. This movement is called translocation. During translocation, the tRNA in the A site moves into the P site. The empty tRNA shifts to the E site and exits the ribosome. A new tRNA enters the A site. This cycle repeats again and again. As the ribosome continues reading the mRNA from 5' to 3', the polypeptide chain becomes longer. Stage 3. Termination. Eventually, the ribosome reaches one of the 3 stop codons, UAA, UAG, or UAG. These codons do not code for any amino acid. Instead of a tRNA, a release factor enters the A site. The release factor breaks the bond between the completed polypeptide and the tRNA. The newly synthesized protein is released. Finally, the ribosomal subunits separate, and the mRNA can be translated again by another ribosome. Clinical importance. Translation is one of the most important targets for antibiotics. Many antibacterial drugs work by blocking bacterial ribosomes and preventing protein synthesis. Examples include Tetracycline, Streptomycin, Chloramphenicol, Erythromycin, Linezolid. Because bacterial ribosomes are 70S and human cytoplasmic ribosomes are 80S, these antibiotics selectively inhibit bacterial protein synthesis. Thank you for watching.